Now that we've talked about proteins, carbohydrates, and nucleic acids, which are all polymers, I want to talk about how polymers are built from monomers via chemical reactions called dehydration reactions, and how they're broken back down into monomers through reactions called through a reaction called hydrolysis. So the way that monomers are built into polymers and the way that polymers are broken down into monomers are actually kind of mirror images of each other in the sense that monomers are put together through a reaction called a dehydration reaction, which involves the removal of a water molecule, and they're taken back apart through hydrolysis, which is a reaction where you put a water molecule back in. So these two processes that we're going to talk about are actually really closely related. And in cells and in living things, we would see these reactions be catalyzed by enzymes or sped up by enzymes. So if we were synthesizing proteins or breaking down proteins, we would have enzymes that were helping us out. So here's a general schematic for a dehydration reaction, which would build a polymer out of monomers. So say we're building a protein, for example, and we've already started to put together our protein. We have our first 3 amino acids in place, and now we want to add a 4th amino acid. That would be our unlinked monomer there. What we would do is we would take the hydrogen from one of the monomers in that short polymer. We would take the hydroxide from the monomer that's by itself, and we would pull them out and turn them into a water molecule. And in that process, what happens is that those two monomers, the third one and the fourth one, get linked together through a covalent bond. So this is dehydration, called dehydration because you're taking a water out, right? Like getting dehydrated, you're removing water, and it results in those two monomers being bound together into that polymer. So I'm going to show you this for each of the 4 categories of macromolecule. So here we have some amino acids that are getting bonded together. So you can see that those purple rectangle shapes are the backbone of they're the part that all of the amino acids have in common. And the yellow and green parts hanging off the side are the R groups. So in each amino acid, when they get bound together through dehydration, the hydroxide group from the carboxyl group of one of the amino acids and the hydrogen from the amino group of the other amino acid get combined into a water molecule, and what is left over is a peptide bond. So the peptide bond, it's just a covalent bond between 2 amino acids. So it kind of marks the place where one amino acid begins and the next one ends. And then here is an image of glucose and fructose, those 2 monomers, getting combined through a dehydration reaction into sucrose, that disaccharide. So glucose has a hydroxyl group on one side. There's a hydrogen on fructose there. They get combined into a water molecule, and that makes sucrose. And in sugar, we call it a glycosidic linkage or bond. I don't really necessarily know why they need fancy names for the bonds between all of the things, but yeah, so the bond that's created has a different name for the different kinds of molecules, but the way that it happens is the same. You take a water molecule out in a dehydration reaction. This is what it looks like for nucleic acids, and condensation reaction is the same as dehydration reaction. That's just another word for it. But so there are 2 nucleotides on the left there. You can see there's a H coming off of the top one. There's an OH coming off of that phosphate group on the bottom, and those are combined to make that water molecule that you see on the right, and it leaves the sugar, the red sugar pentagon of the top nucleotide connected to the yellow phosphate circle of the bottom nucleotide, and that's called a phosphodiester linkage. And then one more example, again, it shows the same thing. So we'll talk about fatty acids and triglycerides in the next video. But for now, this will make sense because it's the same reaction. So here we have glycerol, which is the backbone of a fat molecule, and we have a fatty acid on the right. And what you'll see is that a hydrogen comes off of one molecule, an OH comes off of the other molecule, it makes water, and then those two things end up bound together. So in this kind of molecule, this triglyceride it's called, which again we'll talk about in the next video, you do that 3 times and you get 3 fatty acids to make a fat. Okay, so that was dehydration. So now let's talk about the opposite, hydrolysis. So hydrolysis is the same thing but in reverse. So now you add in a water molecule, you like use up a water molecule, and you split it so that a hydrogen goes to one of the monomers and the OH goes to the other monomer, and those things are no longer bonded together. So hydrolysis adds water, breaks it down into its component parts. And again, it would also possibly be accelerated by enzymes. So here, this is an image of 2 amino acids connected together on the top. So if you look at this carefully on the left, you'll see the blue nitrogen, that's an NH2, that's the amino group. Then the next carbon to the right is the alpha carbon. Then the carbon to the right of that would be the one that had the carboxyl group before they got bound together. Then there's the nitrogen again, and that's where the next amino acid starts. So if you take that water molecule and you give the OH to the amino acid on the left, and you give the H to the amino acid on the right, you end up splitting that chain back into 2 amino acids. So that is hydrolysis. And then one more example, here we have maltose getting broken up into 2 glucose monosaccharides. So we add water and the OH goes to one glucose and the H goes to the other glucose. And so again, hydrolysis broke down that disaccharide into 2 monosaccharides. So here's a summary slide. So dehydration for protein forms peptide bonds. Dehydration for carbohydrates forms glycosidic bonds. Dehydration for nucleic acids forms phosphodiester bonds. Dehydration for fatty acids and glycerol forms ester bonds. Dehydration is the opposite of hydrolysis. So dehydration takes water out and puts things together. Hydrolysis puts water in and breaks things down.